Right now, the world is at a tipping point. Challenges like climate change, plastic pollution, and dwindling resources threaten our ecosystem and future. At the heart of these challenges lies a critical issue, which is waste. In agriculture, vast amounts of organic and inorganic residues are left unused, contributing to pollution, harming biodiversity, and fueling climate change. But what if we could turn this waste into something revolutionary? By harnessing the power of microbes, we can transform agricultural waste into biodegradable plastic, an eco-friendly alternative to petroleum-based materials. This innovation not only tackles the global plastic crisis but also promotes the circular economy, reducing our environmental footprint. Our solution aligns with global sustainable development goals, including responsible consumption and production, climate action, and life below water. It offers a bold, scalable, and impactful pathway for Malaysia to lead the way in sustainable innovation, setting an example for the world. Together, we can turn waste into wealth and challenges into opportunities for the cleaner and greener future. We also referred some government agencies for our project. Microbial technologies offer innovative solutions that align with Malaysia's Sustainable Development Goals (SDGs). The Malaysian government adopted the 2030 Agenda for Sustainable Development (2030 Agenda) together with 192 other world leaders at the United Nations General Assembly in New York on 25 September 2015. This is global commitment towards the more sustainable, resilient, and inclusive development with 17 Sustainable Development Goals and 169 targets. The following are government agencies that are referred for this project. The first one is Ministry of Science, Technology and Innovation. The second one is Ministry of Environment and Water. And the third one is Ministry of Agriculture and Food Security. In alignment with Malaysia's Sustainable Development Goals (SDGs), our innovative project to convert agricultural waste into biodegradable plastics using Ralph's Ternary Propa H16 takes a significant step towards achieving several critical objectives, which is responsible consumption and production, the 12th SDG, the climate action, the 13th SDG, and the life below water, the 14th SDG. Here is the three goals that connected to our product. The first one is a circular solution for responsible consumption and production. The second one is climate action through reduced carbon footprint. And the last one is protecting life below water with biodegradable solution.
As you all know, the rising need for a greener environment and more sustainable methods of development, especially in developing countries, has pushed for the decrease in usage of plastics that are being pushed in the decrease in usage of plastic made from fossil fuels and such, and has pushed for the rise of and the creation of bioplastics. Such materials are hoped to fulfill the same roles as real plastic while also being biodegradable as well as more sustainable to create when compared to real fossil fuel. The issue with such usage of such materials is the production method as well as source materials. Now, our main problem here is that whether or not we can use the techniques of microbiology in the production of the materials from waste products to substitute the roles of plastic, which is what we are talking about, bioplastics. In Malaysia, coconut waste makes up 6.7% of all agricultural waste, amounting to around 80,000 tons a year. Now, in the palm oil industry, 75.61 million tons of solid biomass are generated annually, which is equivalent to around 1.25 tons of waste per hectare of palm oil plantation. The main component of these palm oil biomass waste will be EFBs, fronds, as well as trunks. To add on to all those bio-waste will be rice husks, which are the outer layers of rice grains found abundantly as the byproducts of rice milling industries, which amount to around 770,000 tons a year. not to mention sugarcane waste as well as many other wastes, which also adds on to the annual agricultural waste in Malaysia, in which if they are not properly disposed of, would be detrimental to the environment as well as the ecosystem, as they can attract pests, rodents, cause disease, as well as emit dangerous toxins in the air and water due to degradation.
So for the main issue, which is having difficulties in managing the agricultural waste, we are proposing a solution which is using the microorganisms to decompose the agricultural waste. And the microorganism that we will be using is Ralstonia eutropha H16. The agricultural waste are decomposed and turned into polyhydroxy alkanoates, which were then processed into smaller pellets to create the biodegradable plastics. These can potentially turn any agricultural waste into the useful product that can protect the environment. So for this case study, we developed a modular bioreactor system equipped with multiple compartments and also optimized for different stages of microbial fermentation and product extraction. Ralstonia eutropha, also referred to as Cupria vides necator, Alcagenus eutrophus, Waltersia eutropha, and Hydrogemonomonas, is a gram-negative and non-pathogenic and facultative chemole autotrophic beta-proteobacterium, first isolated from soil approximately 70 years ago. Over the last decades, numerous strains of R. eutropha has been identified, each with distinct metabolic traits that make them valuable for various biotechnological applications. Discovered nearly 60 years ago, R. eutropha H16 has emerged as a model microorganism for the research on lithoautotrophic growth, owing to its unique ability to utilize hydrogen as an energy source during the autotrophic carbon dioxide assimilation. The picture that is being shown now is the fluorescent microscopy analysis of the R. eutropha H16, taken under phase contract and fluorescence conditions with a digital camera called PulseNet, and were processed with the MetaView or Metamorph software, which is called as Visitron system. So the question is, why are we using R. eutropha H16? What is so special about it? So, R. eutropha H16 has a remarkable metabolic versatility, meaning that it can live on a wide variety of carbon substrates, including sugars like fructose, gluconate, organic acids, and even aromatic compounds through heterotrophic growth. R. eutropha H16 also has a robust ability to accumulate intracellular polyhydroxy alkanoates, particularly PHB. And under carbon inhibitor conditions, the organisms can store up to 80% of its dry cell weight in the form of PHB, making it an ideal candidate for the production of biodegradable bioplastic. So, how does our prototype work? First of all, the bioreactor, which is the central cylinder, has a primary function where the microbial fermentation occurs. Here, Ralstonia eutropha metabolizes the carbon source, which is the hydrolyzed agricultural waste, to produce PHB. There are many ports and connections connected to the central cylinder. The top port is for gas exchange, allowing CO2 and methane produced during the decomposition to escape. The inlets allow manual feeding of pre-treated agriculture waste, which is grounded into smaller particles to enhance the decomposition and also the microbial uptake. The outlets enable the sampling, draining, or transferring the culture broth to the other tanks. The feedstock tank, located at the top right tank, which stores the pre-treated agriculture waste, which serves as a primary carbon source for the PHB production. The PHB collection tank, which is located at the bottom right, will collect the culture broth from the bioreactor after the fermentation for downstream processing. Then, the tubing and small tanks, which act as a effluent management and recycling, are the smaller tubing and smaller tanks manage the separation of waste by byproducts, residual lipids, or unused nutrients from the proper disposal or potential reuse. Sterile water or buffer tank, located near the bottom left, will provide sterile water, buffer solutions, or supplemental nutrients that can be fed into the bioreactor as needed. The control system, which is located at the bottom left, has a purpose of monitoring and regulating the fermentation process to maintain the optimal growth and PHB production condition via managing the pH levels, the temperature, and also providing an optimal condition for the fermentation process. In conclusion, the transformation of agricultural waste into biodegradable plastics using Ralstonia eutropha H16 is a groundbreaking innovation that addresses multiple global challenges, including plastic pollution, environmental degradation, and also resource scarcity. By leveraging microbial technologies, this solution not only mitigates the harmful effects of the agricultural waste, but also paves the way for sustainable production practices. This approach aligns seamlessly with the Malaysians' commitment with the Sustainable Development Goals, particularly those focused on the responsible consumption and production, which is SDG 12, climate action SDG 13, and life below water, which is SDG 14. It highlights the potential for integrating science and technology to promote a circular economy, reduce our carbon footprint, and also protect the marine ecosystems. With scalable bioreactor system and modular designs, this project demonstrates how waste can be converted into a valuable resource, setting an example for innovative, eco-friendly practices globally. By embracing such transformative technologies, Malaysia can position itself as a leader in sustainable innovation, inspiring a cleaner, greener, and more resilient future for the generations to come.